In order to meet the demands of constantly changing fashion trends, retailers are producing cheap, low-quality clothing in the industry known as fast fashion. While this allows consumers to buy more clothing more frequently at a lower price, this convenience comes at a cost. The fast fashion industry accounts for 20% of global wastewater, produces 10% of human greenhouse gas emissions, and by 2030 will create more than 134 million tons of textile waste each year across the globe. And this industry is only growing. As of 2020, 52% of clothing produced is made from polyester, an artificial fiber that can take up to 200 years to degrade naturally. The cost of polyester has made it so prevalent that in Canada alone, this fabric is responsible for 7% of plastics in landfills, making it the third largest category of fabric waste in the country. While it may take a shift in societal values to alter the fashion industry, we can reduce the environmental impact of the industry by making informed choices and disposing of our clothes responsibly. As of 2021, only 14% of polyester is recycled within Canada.
The two main methods of polyester recycling are open-loop and closed-loop recycling. Open-loop recycling involves pulling apart textile fibers for reuse, but it results in a decline in quality. Closed-loop recycling involves producing yarn or thread from textile fibers. Methods to manage textile pollution are expensive and generate secondary pollutants. Thus, there is a need for cost-effective and sustainable solutions to polyester plastic waste. Specifically, we wish to target the decomposition of polyethylene terephthalate, commonly referred to as PET. While research has been conducted on the use of naturally occurring enzymes, a system has not been designed that completely digests and metabolizes PET.
We are RHS Calgary, and to combat this problem of fabric waste, we present our solution, FabriFuel, an innovative and refreshing approach designed to address fabric waste while promoting sustainability. We aim to address this problem of fabric waste by designing a microbe capable of digesting polyester as its primary energy source, while harnessing the net production of electrons using a microbial fuel cell. Our research has two specific objectives. We aim to, 1, design a bacterial system capable of fully digesting PET and its byproducts, and 2, capture the energy produced as a byproduct of the cell's metabolism. It is our hope that the production of energy from these microbial fuel cells will incentivize adoption of these cells and make them more cost-effective. In this, our product promotes both environmental and economic sustainability by reducing harmful wastes in our environment and producing greener, cleaner energy. Currently, there is a lack of cost-effective and sustainable ways to recycle polyester and other plastic-based textiles, which leads to waste and the creation of toxins. Recycling methods are also not promising, as recycling polyester requires many harmful chemicals, including chlorine, and results in the release of microplastics that can further harm the environment. Furthermore, our research addresses current gaps in knowledge, such as effective pathways for the microbial digestion of PET. The first aim of our project is to design a microbial system capable of completely degrading PET into usable metabolic byproducts. The first question we aimed to address is how we can utilize an enzymatic system to break down PET into byproducts. We began by extensively searching literature for bacteria capable of digesting PET. One such bacteria is an aerobic bacteria found in aerated soils, Ideonella sakaiensis, which secretes two enzymes, which together are able to break down PET into its monomers. This two-enzyme system consists of metase and petase. In this reaction, the petase enzyme attacks the polymer PET, liberating bis-hydroxyethyl terephthalate, known as BHET, mono-2-hydroxyethyl terephthalate, known as MHET, and terephthalic acid, TPA. From this, petase will further cleave BHET to produce more MHET and ethylene glycol, referred to as EG, and MHET is hydrolyzed by metase into TPA and EG. These reactions were identified by previous research done by Dr. Grade Beckman's lab in Colorado. Their studies have shown success in expressing a chimeric protein that combines both PETase and METase from a single plasmid. This chimeric protein is found to be more efficient in its interactions with MHET. Thus, we chose to incorporate it into our system. We determined that the first stage of our project should include BL21 E. coli bacteria, which is suitable for high-efficiency transformation. Transformed with our plasmid, coding for the secretion of the METase-PETase protein, thus breaking down PET into its primary byproducts, terephthalic acid and ethylene glycol. The second question we aimed to address was, how can we break down the byproducts of METase-PETase to be utilized by the cell? In order to break down TPA, we need to incorporate the genes for carboxylic acid reductase, which converts TPA into usable aldehyde forms. To break down EG, we would need to overexpress two enzymes, propanylyl oxidoreductase and glycoaldehyde dehydrogenase. However, upon speaking to Dr. Laura Keffer-Wilkes from Lethbridge University, we recognized a flaw in this solution. Our E. coli will not be able to take in a large plasmid, and it is difficult to transform E. coli with two plasmids. So, we decided that it was best to utilize multiple bacteria for the digestion of TPA and ethylene glycol. However, our bacteria containing multiple plasmids cannot exist in the same environment, as they will conjugate and exchange plasmids, which we do not desire. Thus, we generated the idea of a multi-chamber system separated by semi-permeable membranes that allow for each reaction to take place and for PET to be entirely degraded. Our first chamber will include the BL21 bacteria transformed with our METase-PETase protein. The reactant of this chamber will be our polyester, PET, and the products will be TPA and EG. Knowing that E. coli is approximately 1 to 2 micrometers long, we aim to use a semi-permeable membrane, such as dialysis tubing with a specific pore size, to allow for only our desired products to move to the next chamber. Considering the size of both ethylene glycol and terephthalic acid to be smaller than the E. coli, we intend to use dialysis tubing with a pore size that is smaller than 1 micron. This will allow EG and TPA transport across the membrane. The second chamber of our bacterial process involves the breakdown of TPA into a form that is easily metabolized by the bacteria. We identified the ideal enzyme for this process, carboxylic acid reductase, on the premise that it is shown to be successfully tested in E. coli, where the products of its digestion were easily metabolized by the cell. By introducing a plasmid containing carboxylic acid reductase into our second E. coli, TPA will be converted into two aldehydes, such as shown on screen. There is evidence that these aldehydes will be naturally degraded by endogenous enzymes in the E. coli. Thus, the reaction in the second chamber involves breaking down TPA into usable aldehyde forms. The final chamber of this process will break down ethylene glycol. E. coli is only able to degrade EG when the two enzymes are overexpressed, propanylyl oxidoreductase and glycoaldehyde dehydrogenase. Thus, we have two ideas for genetically modifying our third bacterium to utilize EG as its primary energy source. Number 1, directed evolution, and number 2, introduction of a plasmid coding for the enzymes. By introducing our bacteria to multiple rounds of UV radiation, we hope to encourage mutations. The bacteria will be exposed to high levels of ethylene glycol, and the most robust colonies will be selected for the next round of experimentation. This is our third research question. How can we engineer bacteria without the use of plasmid transformation to digest ethylene glycol? If we do not find success with UV radiation, we may utilize ethylmethane sulfonate mutagenesis utilized by researchers from the University of Barcelona. This is the final reaction in our process, wherein our third bacteria will be able to completely digest ethylene glycol. Thus far, in our experimentation process, we have completed a few key steps. 1. We have created chemically competent cells, which allow us to ensure that our target plasmid DNA can be harnessed and cloned. 2. We have transformed our enzyme of interest, the METase-PETase chimera, into DH5 alpha. 3. We ran mini-prep and maxi-prep procedures in order to isolate plasmid DNA from bacteria and are currently quantifying the concentration of DNA in our samples using a nanodrop. 4. We have completed restriction analysis to fragment DNA sequences with specific endonucleases and have conducted gel electrophoresis to map our DNA fragments. Our future experimentation involves expressing and purifying our chimeric PETase-metase transforming bacteria 2 with a plasmid containing the gene sequence for carboxylic acid reductase, and directing the evolution of bacteria 3 using mutations. Upon confirming the reactions in each of the chambers with our 3 genetically modified bacteria, we will test the optimization of factors such as the growth medium and temperature, and will determine how to connect the chambers to make a cohesive system. Other experiments we aim to complete are listed on the screen.
The second objective of our project is to generate electricity as a byproduct of the PET degradation reactions. A microbial fuel cell works like a battery, capturing electrons from biochemical reactions in bacteria to create an electrical current. There are 2 halves separated by a semi-permeable membrane, which allows hydrogen ions to pass through. Each half contains a conductive surface to facilitate the transfer of electrons. One of these surfaces acts as the anode, also known as an electron acceptor, and will be placed with the bacteria. The other will be the cathode, the electron donor, and will be exposed to the oxygen in the atmosphere. The production of electrons is a key part of the bacteria's metabolical process. The microbial fuel cell takes advantage of this by acting as an acceptor of electrons. The electrons that gather on the anode will flow to the cathode, resulting in an electric current. These are the basic principles of all microbial fuel cells. Our microbial fuel cell uses graphite for both the anode and the cathode, as it is not toxic to bacteria and readily available. We use a commercially available material for the semi-permeable membrane, as well as an acrylic housing container. The graphite plates are designed so they can be layered together to create additional repeated chambers for each of the following half reactions.
The 2 reactions that occur are O2, oxygen gas, plus 4H+, hydrogen ions, plus 4 electrons, goes to 2H2O, water, at the cathode, and C6H12O6, glucose, plus 6H2O, water, goes to 6CO2, carbon dioxide, plus 24H+, hydrogen ions, at the anode. These half reactions allow us to develop predicted electrical potential values for the fuel cell. The theoretical electrical potential of the cathode reaction is negative 0.43 volts, and the theoretical electrical potential value of the anode is 0.51 volts, which gives us an electrical potential difference of 0.94 volts for the total electric potential value of our reactions. It is important to note that these equations assume STP, a pH of 7, and standard atmospheric concentration of oxygen.
As of right now, preliminary testings has been completed, and we have developed a proof of concept to make sure that our microbial fuel cells components work properly and to identify variables that could influence the outcomes from our microbial fuel cell. For further experiments, we would like to test the system with the modified E. coli in the microbial fuel cell in order to determine if the modified E. coli is as electrically active as the unmodified version. In the future, we hope to address one primary question. How can we combine both the 3-chamber bacterial system with the microbial fuel cell to create a cohesive system?
Our third aim is to create awareness regarding the problem of fabric waste and to connect with key stakeholders who will allow us to better target our project. While the wet lab and dry lab portions of our team have been working closely together to bring this project to fruition, our human practices team has been working to fulfill 2 objectives. 1. Identifying and speaking to relevant stakeholders to assess the relevance of our project in the real world, and 2. Raise awareness and understanding of the issue of fabric waste. The HP team has contacted a number of individuals in the areas of textiles, plasma design, and in microbial fuel cell technology. We have faced a challenge in finding stakeholders to speak to. Despite this challenge, we have created interview guidelines for future stakeholders. Furthermore, we have found a few mentors who have aided significantly in our project. Robert Mayo and Emily Hicks, the co-founders of FredSense, have been our leading mentors in supporting us with the development of our project. They helped us prevent and circumvent potential roadblocks in our prototype and gave us unconditional support on how we should best develop our idea. To fulfill our second objective, we have designed a survey meant to understand the current knowledge of fabric waste. Through our project, we ultimately hope to spread awareness of the important issue of fast fashion and fabric waste, and to utilize our project to incentivize individuals to invest in sustainable clothing and fabrics.
Finally, we would like to acknowledge the help of our mentors, Dr. Yasi Suarez, Ms. Dallas Mithril, Ms. Alyssa Crispin, and Ms. Kim O'Feef. They have unconditionally supported us both through the highs and the lows.
The issue of fabric waste is prevalent, and it is crucial that it's addressed. We aim to develop our project enough to implement it into a real-world context. Ideally, we would like for textile manufacturers, distributors, and recyclers to use our product to sustainably dispose of polyester waste. We hope our project will offer a better alternative to current methods of recycling polyester, which are both harmful to the environment and not economically friendly. We, as consumers, enjoy choice in what we wear. But oftentimes, we don't see the immense negative effects of these choices. Fast fashion, a rapidly growing industry in our world, has been recklessly encouraging wasteful consumer choices. With its growing prevalence, there comes a wide array of environmental issues stemming from fabric waste, which, if not addressed now, will only continue to drastically affect the world we live in through increased greenhouse gas production, excessive water depletion, and much more. It's important, now more than ever, that we take action and begin to solve the problem of fabric waste. We are RHS Calgary, and we created FabriFuel, an innovative approach to solving this problem of fabric waste. FabriFuel, reducing waste, producing energy, all in a single solution.